Wildlife disease is a growing issue for conservation. Currently, 4% of global species declines are due to disease, but this number is likely to increase as humans are spreading pathogens to new environments and naive host populations at unprecedented rates. One example of such a disease is white nose syndrome, caused by the invasive fungal pathogen Pseudomonas destructans, or PD for short. Since the introduction of PD to North America, white nose syndrome has killed millions of bats across the continent. It has caused regional extinction of some species, and it has resulted in the fastest mammal decline in history. Now, part of the particular reasons for these dramatic population declines is PD's ability to survive and grow in hibernation environments like caves and mines in the absence of their bat hosts. This allows the fungus to colonize these areas and quickly infect healthy individuals when they enter infected caves for hibernation. And although bats can survive the summer and get rid of the infection, many don't survive the winter to do this. So my research is specifically looking at using ultraviolet light and a chemical called PEG in treating the environment. And the reason we're going to do this is that if you treat the environment to get rid of the fungus, PD, maybe we can push back the initial infection date that bats get infected, which will lower the fungal load and the disease severity. We might also reduce the rates of infection between bats and the environment, ultimately reducing mass mortalities. So what I am doing is I am treating 3 abandoned mines at the northern and southernmost frontiers of PD spread with ultraviolet light and PEG before the onset of hibernation. I'll also be swabbing the mine substrates, so the walls and ceilings, which will allow me to determine the effectiveness a one-time treatment of UV and PEG has over the hibernation season. I'll also determine the effects on other microbes in these areas, which might be important for hibernation environments or for bats themselves. Results from my research will be able to inform management planning for environmental cleaning as a white nose syndrome management strategy, and treatments might be able to be a long-term solution to promote survival and maintenance of bat colonies. Treatments also might occur on the frontiers of PD spread, which could protect environments not yet infected. Results from my study, my study is the first to test for treatments for PD in the environment and may be able to provide insight to management of other wildlife diseases. Thank you.